Welcome to AP Biology. This presentation is going to be an overview of meiosis. So the first thing that happens when we go through meiosis is we're going to have condensation of the chromosomes, just like we had during mitosis. And the nuclear membrane is going to dissolve and the spindle fibers are going to form their spindle fiber network. It's all kind of similar stuff to mitosis. But the difference is the homologous chromosomes are going to form tetrads and wrap around each other and swap a little bit of DNA. And so that's the first major difference. The next major difference is that in meiosis, instead of having individual chromosomes lined up along the central plate, you have pairs of homologous chromosomes lined up along the central plate. And so when we go through that first anaphase, instead of pulling sister chromatids apart, we're pulling whole chromosomes to the opposite ends. And then the second part of meiosis is almost identical to mitosis. You have single chromosomes lined up on the central plate, and you pull sister chromatids apart just like you do in mitosis. All of the cool stuff that takes place in meiosis takes place in the first division. So, crossing over, which is super important, is the first thing that happens. And we have swapping of genetic information from the maternal and paternal chromosomes, the one from your mom and the one from your dad. And then the chromosome pair will line up on the central plate, but they will do it independent of one another. And so whether red is facing one way or blue is facing the other is independent of the other chromosome. We call this independent assortment. There's a law of independent assortment, and it basically just means that the chromosomes are going to line up independently during meiosis. And then you have one of the two chromosomes pulled to the opposite poles, and we call that segregation. Segregation means that your chromosomes are separated from one another during meiosis. So we have a law of independent assortment and a law of segregation. One says the chromosomes are separated during the first part of meiosis. The other says that they get separated independently of one another. The second part of meiosis is basically just a copy of mitosis. The chromosomes line up on the central plate during metaphase, and then they get pulled apart during anaphase, and then we get nuclear membranes form, and the chromosomes decondense just like they do in mitosis. And you end up with your 4 gametes. Okay, so those gametes are going to be the sex cells. So the differences between mitosis and meiosis. In mitosis, we only have 1 division. In meiosis, we have 2. In mitosis, we line the chromosomes up individually during metaphase. And in meiosis, we line them up together in the first division and then individually in the second division. In mitosis, we end up with the correct number of chromosomes in the final cells, because we're making body cells, so we want to have the normal number of chromosomes. In meiosis, we end up with only half the normal number of chromosomes, which is what we want because we're making sex cells. They're going to unite with another sex cell and get back to the normal number of chromosomes. So basically, meiosis is there to reduce the number of chromosomes and mix things up. Okay, we mix up the genes on the chromosomes, we mix up the chromosomes, we reduce the number of chromosomes so that when we make babies, there's lots of variation and the normal number of chromosomes in the offspring. So why have this process where we go through all this work to produce genetically mixed up organisms. Well, in our next video, we'll talk about how variation can help organisms be successful.